Good morning, good afternoon, or good evening, depending on from where you're joining us. It's with great pleasure that I welcome you here today for this webinar entitled Fast Raster Scan Optoacoustic Microscopy Enables Assessment of Human Melanoma Microvasculature in Vivo, based on work that was recently published in May of this year in Nature Communications. My name is Neil Burton. I'm the Vice President of Applications at iThera Medical, and I'll be moderating this session. The presentation itself will last for approximately 30 minutes, and we really do encourage questions for the Q&A session at the end. If you maximize the GoTo tool, you can see that there is a question box where you can type in your question, and then at the end, we'll address those questions. The webinar will also be recorded, and we'll send a link to the recording to you over the next few days by email. So our speaker today is Dr. Hailong He, and he's the group leader of the optoacoustic microscopy group at the Institute of Biological and Medical Imaging at the Helmholtz Center in Munich. He did his PhD in the Faculty of Electrical Engineering at TUM. His focus is on the development of novel imaging systems like optoacoustic endoscopy and raster scanning optoacoustic microscopy, which we call RSOM, and he's engaged in biological and clinical studies. His work has pushed forward applications of optoacoustic imaging, particularly in the clinical space, including human melanoma detection and assessment of microvasculature complications of diabetes and cardiovascular disease. In this talk, he'll introduce the recent development of RSOM and new clinical applications. And with that, I'll hand it over to Dr. He.
Thank you very much. Thanks for the kind introduction. So hi everyone, my name is Hailong He. So today I'm going to present to you the recent development of the acoustic microscopy, which allow us to assess the human melanoma microvasculature in vivo. So for today's presentation, the general outline, including these 4 parts, that I will first briefly introduce of the acoustic imaging for those who are not very familiar with this technology. And then we are talking about the development of a raster scanning of the acoustic microscopy, and then a single breast-hold RSOM for melanoma imaging, and then at the end I will summarize, the work we have I'm presenting. So, of the acoustic imaging, maybe you guys are more familiar with ultrasound. For instead of sending sound of the acoustic imaging, we're sending pulse laser light. The light hit the tissue absorb the energy, and energy express an ultrasound wave. And then we use an ultrasound transducer to record the waves. So because of this, we have this unique property. So this modality has the optical absorption contrast and with ultrasound resolution. So then, since the sound is scattering much less in tissue compared to light, so we can penetrate deeper compared to pure optic approaches. So here, for example, with the implementation of the acoustic microscopy, we can resolve the 3D microvasculature of human skin with a resolution in the range of 30 micron laterally, axial resolution around 10 micron. And then we can penetrate around 3 to 5 millimeter deep inside tissue, depending on the wavelength. And of course, also we can implement in tomography mode by sacrificing the resolution. For example, here shows a cross-section image of a mouse body. We can achieve 150 micrometer resolution and around 5 centimeter penetration depth. This image was acquired by the MSOL system from iThera. Another important feature of optoacoustic imaging is the multispectral capability. So because the tissue can absorb the light is dependent on the wavelengths. So by using multi-wavelengths, we can excite the tissue, and based on the tissue absorption spectrum, we can actually first to selectively contrast at different wavelengths. We can highlight the specific chromophores based on the absorption peak, and also we can resolve the concentration of tissue components. Here shows, for example, using 4 wavelengths, we can resolve the melanin, oxyhemoglobin, and deoxyhemoglobin of the human skin structure. And also, using this iThera Acuity MSOL system, we can actually, for example, here shows that you can resolve the oxy, and lipids of the human breast. So then comes to the optoacoustic microscopy. So I think most of you are more familiar with microscopy, like optical microscopy, which can provide very high resolution in the range of a few micron, even down to sub-micron, and with penetration depths in the range of a few hundred micron up to 1 millimeter. And then there's also a microscopy like the tomography system I just mentioned, so which can achieve a resolution in the range of above 150 micron, and then with the depths above 1 centimeter. But between microscopy and macroscopy, so we define a region called mesoscopy. So this modality can achieve a resolution in the range of tens of micron and the penetration depths from a few millimeter up to a centimeter range, depending on the wavelength. So this is very suitable for imaging the human structure, human skin. Because of the human skin, it contains a layer structure. It has an epidermis and dermis, and also deeper into the muscle layer. And also the skin thickness is varies from several millimeter up to centimeter range, depending on the location. And also in the skin, for example, in the dermis layer, they contain vessels. The vessels, with a diameter range from tens of micron, like the capillaries, and go to deeper, you have larger vessels up to more than 100 micrometer diameter. So that means this need requires an image modality, which has high resolution to resolve this structure like the vessels, and also to have enough penetration depths to image through the whole depth of the skin. So this mesoscopy system is ideal tool for skin imaging. That shows the first generation of the raster scanning of the optoacoustic mesoscopy we have developed in our institute. So this is a schematic of the system. It has a 2 fiber bonder to eliminate the tissue. We have a ultra broadband transducer, which can record a frequency from tens of megahertz up to 120 megahertz. This super broadband width, it allows us to achieve high resolution, because you may know, the resolution is determined by the bandwidth of the transducer. And here shows, this is the picture of the scanning head. And here's the first image. We resolved the cross-section image of the human skin. We can see the epidermis and dermis, and also in the dermis we see the capillary loops layer and the vascular plexus. So if you look at the maximum intensity projection from the top view, we see the epidermis, the capillary loops. We see this dot because the vessel is vertically visualized. So we measure like the dots. And here the vascular plexus, we see the vessel network very nicely. This is for the first time there's an image modality and non-invasively to resolve the structure of the skin at such high resolution and the deep penetration depths. Because here we're using a green light, so we achieve the penetration depths around 1.5 millimeter. And then with this device, our institute tried the first application is for the psoriasis skin. So we measure the psoriasis skin and compare with the healthy skin. We can here's a cross-section image, and we clearly see there's a morphological difference between the psoriasis skin and healthy skin. For example, we see this elongated epidermis and also dilated vasculature compared to the healthy skin. Furthermore, recently we have applied a study to using RSOM to monitor the treatment and to quantify the changes during the treatment of psoriasis. Here first row shows the psoriasis skin on different days with the treatment. Is this PASI value? This is a clinical score to quantify the severity of the disease. So here's the cross-section image of the RSOM. So with the higher value, that means the severe of the disease. So for example, the PASI 7, the first day of the of the treatment, we see this very thick epidermis. This is this the thickness. And then with the treatment, we will see this feature was changed. So we see the epidermis is get narrowed, and also the vasculature, also the features of the dermal vasculature, we also see the changes. So by monitoring those features, for example, the epidermis thickness changes, we can quantify the treatment of psoriasis. So if you look interested into this study, you can go to this publication. And besides single wavelengths, we also actually do multi-spectral RSOM by using OPO laser. We can actually to highlight a different chromophore skin. For example, we're using 515, 532. This more focus on the hemoglobin in the vasculature. And then if you use a 650 nanometer, we see the signal mainly come from the melanin. 1210 nanometer, we see subcutaneous fat, the subcutaneous glands, and water. And then 1450 mainly is the contrast comes from water. And there shows this nice composite images. And then we come to, for example, to discuss about the possible improvement in RSOM. This study we just mentioned, we have one of the issue is the speed, because we have this laser safety limit. So this requires us to to reduce the repetition of the laser, which will slow down the scanning speed. For the first generation of RSOM, and normally it took like 30 seconds to scan or the single wavelengths, for example, the green light 532 nanometer with a maximum repetition around 500 hertz. So this only allows us to image, for example, the arm or legs, where have much less motion from the breathing. But if you try to measure, for example, the body trunk, like and also the head region, there's like a very strong or motions. This will kill the quality of the images. Another thing is for the spectral. So we want to further to expand, for example, the multispectral scheme of the RSOM. But this requires also we need to get a faster laser source, which allow us to do faster multispectral imaging. And this can help for them to resolve the functional molecular information. Yeah, it comes to, for example, the motion, the effect of motion. Here shows an example. Here, for example, if we fix our ultrasound detector and then we measure the signal, we can see this up and down motion generated by the pulsating of the artery. This motion will destroy this continuity of the vasculature. For this, as this is schematic shows. And here shows, for example, if you look, fix our detector on this, on the position of the skin tissue, we see two types of motions. Here shows this is the strong motion within the range around 100 micron, and here it is we see some more like in high frequency motion in the range around 50 micron. So this motion we can actually correct it by using motion correction approaches. But when you trying to measure the skin at the body trunk, for example, especially close to the heart region, this breathing motion are very strong. They are in the range of around more than 300 micron. So in that case, there's no way to correct those motion. And why we are slow? Why the RSOM, the first generation, is a slowdown? Or why there's limit? There's main the main limitation, as I mentioned, is because of the laser safety limit on the human skin. This is determined by several factors like the illumination size, wavelengths, illumination period, and also the repetition rate, the laser source. For example, for this our first generation of the RSOM, we use a two-fiber bundle, which will illuminate around 5 millimeter diameter circle. And actually, our detector, like this shows here, this the sensitivity field of detector. So that means a lot of the light are waste because this signal from here are not recorded by our detector. So then we come to a new configuration, this coaxial illumination. So we actually reduce the illumination area. So this will just barely cover the sensitivity field of the detector. In this case, we will significantly reduce the illumination area of the skin, and then we can continue to increase the repetition rate. For example, we're using 1.4 millimeter diameter illumination spot. And then we can continue to increase this repetition rate of the laser source up to 1.5 kilohertz. So that's we can increase the scanning speed at least 3 times. So with this new configuration, here shows this implementation of the first RSOM scheme. This is the ultrasound detector, similar to the first generation. The difference is we drill a tiny hole with a diameter in the range of 300 micron, and then we use a single fiber to transmit the light. And then this can allow us to achieve a coaxial illumination and detection. And with this small illumination spot, as I said, so we can significantly increase the repetition rate of the laser source. Here shows some example of this system. Here we measure the microvasculature of the nail fold. We see these capillaries, and here shows with this same configuration, we're using a fast laser source with 4 wavelengths. We can actually quantify oxy, deoxy, and melanin. Here shows the cross-section image, but the 3 color indicated the different chromophores. If you switch off the melanin signal with this only the hemoglobin left, so we can highlight the contrast from the melanin. This from the top view, the maximum intensity projection. And here we did we did a comparison, the conventional, the first generation of RSOM and this first RSOM configurations. So as I mentioned, so with these two boundaries, we have a larger illumination spot. With this single coaxial illumination, we have a much smaller spot. And also for this transducer, we actually do an improvement by directly touching a preamplifier. So this will allow us to amplify the signal directly and also to provide a good impedance match. This will reduce the noise level of the signal. Here's a comparison between the through-hole transducer and the conventional transducer. We achieve like a 2 times increment of the SNR. Here's a comparison of the skin imaging by these two systems. We measure the same region of the skin. Here, so we can see the vasculature, the similar vasculature was resolved in both modalities. Here is from the conventional RSOM, the first generation, and this is from the first RSOM. And we can see get very comparable quality. So that means by even reducing the illumination spot size and also by increasing the repetition rate, we still get same image quality, and also the penetration depth is the same. And then we're trying to do to measure on the body track. So this we implement this as single breath hold. So we ask the volunteer or patient to hold the breath. Here shows example. For example, if you fix the detector in the back region of the volunteer, and even then in the normal breathing condition, we see this strong breathing motion. This dashed line indicates the motion, this up and down motion. And then if you simply hold the breath, we can see this is significantly reduced. Here's this computation of the motion profile. So this generates the motion in the range of around more than 300 micron. While when you hold the breath, we see the motion has been significantly reduced in the range below 100 micron. I mean, in this case, in the normal breathing motion case, even we apply a motion correction code, we cannot record the image quality of this scan. But when they hold the single hold the breath, we by applying the motion correction code, we can get high quality images of the skin. So we also demonstrate the capability of the fast RSOM by measuring a psoriasis lesion on the patient's back. Before our first generation of the RSOM, we only can measure the psoriasis lesion at the on the leg or on the arm region. So this is the first time we measure this lesion on the back, and we can resolve the similar property of a psoriasis. We see this elongated epidermis, and also compared to the surrounding healthy skin, we see clear difference. And then when it comes to another important application is for melanoma imaging. So as you may know, the diagnosis of melanoma right now is in clinics mainly relies on the dermoscopy and the histologic analysis of the excised lesion or biopsy. And then this is, the visual examination is very subjective, and also the accuracy depends on the, the experience of the dermatologist. And there are many false positive and unnecessary biopsy. So this is a huge need to get a non-invasive imaging, which can assist the visual inspection for melanoma detection, for example, by resolving the lesion microvasculature. I mean, there are actually some other modalities has been used to resolve the vessel structure of a melanoma. For example, dermoscopy, which can resolve the superficial the vessel structure. OCT angiography, this is the projection image. And as we know, so for the pigment lesion, OCT angiography can only resolve the vessel depths up to 500 micron. So then when it comes to our RSOM imaging, so we have tried for the first time to measure the melanoma from a patient's back. Here's a picture of the lesion. So we scan 3 regions. One is in the center of the lesion, one is on the edge, and another one, the third one is on the surrounding healthy skin. The first row is the cross-section image, and here is our maximum intensity projection from the top view corresponding to the epidermis and the dermal vessels. So here we see the pigment distributions inside the lesion, in the edge, and in the surrounding healthy skin. And more importantly, we see the microvascular patterns. For example, inside the lesion, we see this irregular and dense pattern compared to the surrounding healthy skin. We see a clear difference. And here shows a 3D, a volume of a nevus from a healthy volunteer on the back. And here is the stitch image of the melanoma from the healthy skin and the melanoma. We can clearly see how the vasculature involves from this irregular network from the healthy skin to the lesion center becomes irregular and dense. And here shows this the 3D of the 3 scans, 3D volume of the 3 scans in tumor center, tumor edge, and healthy skin. So from the 3D volume, we also see there's a difference of the vasculature. And then we do a comparison study. We actually measure like 10 patients of benign nevus and 10 patients of a melanoma. We acquire 17 lesions from both groups. The first row is from example of a nevus, and the second row is from a melanoma. And here's a cross-section image. And then if you go look at the dermovasculature, especially in the surrounding region, because we mainly measure the edge part of the covering the lesion and the part of the surrounding healthy skin. So if you look at the edge, the vasculature in the edge of the nevus lesion, we see a very regular network close to the healthy skin. While if you look at this the vasculature at the edge of the melanoma, we see this in irregular dense patterns. So actually, and also if you compare these two groups, we see clear difference, for example, in terms of total blood volume, vessel density, tutorosity, fragment lacunarity. So these all these features can use to characterize the density, complexity of the vascular network. We see a very obvious difference between these two groups. So this suggests a potential by resolving the vasculature of the melanoma, it can help to detect, the melanoma non-invasively. And furthermore, we actually also implement a dual wavelength fast RSOM. So we're using two wavelengths, 515 and 532 nanometer. And by using these two wavelengths, we can differentiate the melanin and hemoglobin. So for this two wavelength scan, it took about around 30 seconds. So this cannot do a single breath hold, unfortunately. Here shows, for example, like the image from 515 and 552, and then this two colors to highlight the melanin and the vasculature, hemoglobin. Sorry. And here is more clear. For example, for this melanoma lesion, we see the pigments in the epidermis region, and also the vasculature in the red color in the demo vessels. And here is another lesion, melanoma lesion. And here is with this separation of melanin and hemoglobin, it can actually helps to assess the margin of the lesion. So now I can come to like the summary of this work. So for this study, we actually developed this fast RSOM, which allows to do single bristle scanning, enables to visualize skin structure, from the whole body, like the body trunk, head, and also the arm, leg region. And also this fast RSOM allows to resolve the vasculature of melanoma at the patient's back for the first time. And then we, from the RSOM image, we see clear difference between the malignant and the benign lesions. And then also we implement the dual wavelength fast RSOM, which can resolve the melanin and hemoglobin. This can potentially be used for the lesion margin assessment. So at the end, I would like to acknowledge first to thank our institute director, Professor Nito Christos, and also my group member and collaborators. Of course, also the founding source. Thank you very much for attention. I'm opening for questions.
Okay, great. That was brilliant. Thank you very much, Dr. He, for a really nice talk. We have a lot of great questions, so I'll just start out. But if you do have a question and you haven't yet typed it in, you can do that in the questions tab of the GoTo interface. So the first question here is, maybe I'll start there because it's connected to what's still on the screen. You show Winther and Inoderm as funding projects. What's the future for this project? Is it still ongoing? Is it finished? Where's it going?
Yeah, this Inoderm is actually just finished. So this project is mainly focused on the development of RSOM for disease detection. And the winter is ongoing project. Actually, for this melanoma study is funded mainly by the winter is in the scope. So winter is mainly we are focused on the therapeutic monitoring, like to assess the treatment performance. So by applying an RSOM, we're going to study the microvascular changes, for example, in the patients with diabetes or like cardiovascular disease. So we're going to monitoring the morphological changes and also the function changes by using RSOM. Yeah.
Great. Next question. It says in your paper, you've quantified things like tortuosity, fractal counts, and lacunarity. For this, what software did you use? And are these image analysis approaches something that others could incorporate into their image analysis? So in other words, is the code and or software available to others?
Yeah, yes. All this method to categorize the features of the images are available online, and also these are actually existing approaches which has been widely used, for example, from other medical imaging modalities like the CT or MR. So yeah, so you can find those approaches, from the paper clearly explained, and also the online source for the code. Yeah.
Great. This next question is more melanoma specific. Says, was there a difference in oxygen saturation between a nevus and a melanoma? Yeah,
actually, that's a great question. We are actually, unfortunately, right now we haven't studied that yet because in order to compute the oxygen saturation, we need to use several wavelengths. As I mentioned, for example, if you're only using 2 wavelengths, there's a way we can compute the oxygen saturation. But with 2 wavelengths, we still cannot do like a single breath-hold. For example, it means we cannot finish the 2 wavelengths scan in 15 seconds. So this restricts our capability for melanoma study. And we are working on that. Actually, we're further developing this RSOM to be, in terms of the speed and especially. So yeah, this is an ongoing study we are doing right now. Besides assessing the microvasculature, we're also very interested to see the oxygen saturation changes. Yeah.
Thank you. The next question is about the breath holding, which you were just talking about. It says this has been used in fMRI, near-infrared spectroscopy, and laser speckle to study changes in perfusion. And in these cases, or in your case, breath holds improved image quality in your study, but what impact do you think breath hold has on the underlying biological processes that you want to measure?
Yeah, for our case, simply because because the melanoma or those nevus lesions, a lot of them located in the body trunk. So by holding the breath, this allow us to significantly reduce the motion, which can help us to recover the image quality. So this is a very obvious improvement, as I mentioned in the slides. Yeah.
Okay, good. Next question. You showed an ROC curve for melanoma where you compared blood volume to tortuosity to blood volume plus tortuosity, and it was the combination of the 2 parameters that gave you the best outcome. So how did you combine these outcome measures to improve sensitivity?
Yeah, this is actually also a very standard approach called multivariate logistic regression. So it's by combining different parameters to compute this AUC ROC curve. Yeah, this also I have a clear explained in the paper. So if you're interested, you can also to go to seek for the details there. Yeah.
Okay, great. I don't see any other questions at the moment. So I think I would therefore close the webinar. If you do have any other questions, you can certainly follow up by email and we'll get an answer to your questions. And we will soon post this webinar on YouTube and follow up with you all with the link in case you'd like to share the webinar. Thank you again for your attendance, your attention, and also your questions, and have a nice day.
Thank you.